Hello, dear students. Welcome for our next lecture on cell and molecular biology. As usual, we will go to study the prokaryotic and eukaryotic cells. Unit number 2. In first unit, we have seen the basics of cell biology and molecular biology. So, what is cell biology and what is molecular biology? We have studied at primitive level that the structure, its components, its working, its functions, and the molecular biology we have seen DNA, RNA, and their types and their role in the cell life or cell activities. Now here the prokaryotic and eukaryotic cells are the two basic type of cells in which the prokaryotic cell is the cell having nuclear material but not bounded by the nuclear membrane, and in eukaryotic cell the nuclear material is bounded by a nuclear wall or nuclear envelope, and therefore these are the real or true cells, for example the plant cell or animal cells. So in this topic, unit number 2, we have to study these 4 topics basically. Of them, first is general organization of prokaryotic and eukaryotic cell, or we can say typical animal cell. As we are the students of geology, we have to study the animal cell. So what is the general organization of prokaryotic and eukaryotic cells? So all the cells are membrane bounded units of living matter, and these are of two basic types, that is prokaryotic and eukaryotic. So differing considerably in their structural organization, the prokaryotic cells are very small and structurally very simple, or we can say primitive type of cells having distinct nucleus. These mostly exist on earth as free unicellular organisms and include mycoplasma. Bacteria and blue-green algae, or we can say cyanobacteria, are the best example of unicellular prokaryotic cell types. In other hand, eukaryotic cells are larger, structurally complex, and are nucleated cells. Means nuclear material is bounded by special membrane and so called as nucleus. In prokaryotic cell, the nuclear material is scattered randomly throughout the cell or matrix, and therefore we can call it as a prokaryotic cell. Next, what is the ultrastructure of these all the cells, either prokaryotic or eukaryotic? Actually, we have discussed these all the topics in our previous lecture when we have studied what is the basic cell types. In that, we have seen that the cell material, any type of cell material, is generally called as matrix, or if there is a separate nucleus, the remaining material is known as cytoplasm. Or in simple way, we can call it either cytoplasm or matrix, or together whatever the material is present in the cell is generally known as protoplasm, to which we can divide as cytoplasm and nucleoplasm. So in spite of these all the things, the cytoplasm is the material excluding nucleoplasm. It contains various cell organelles, mitochondria, Golgi bodies, endoplasmic reticulum, plastids, etc. as per the type of cell, either plant or animal, and they work as a machinery, or we can say they work as a special predictable system, and that work regularly, continuously for the development and propagation of the cells. So they perform various biochemical activities and metabolic activities in a cell with a systematic manner, which is organized by the organ or system. Secondly, some organisms are made of only single cells, as we have studied already in bacteria or blue-green algae, and are the unicellular organisms. We can say unicellular cell as well as organism. Like most of the protozoans are unicellular organisms, and they are studied basically as a typical animal cell type or animal single cell type. These cells are fundamental and basic building blocks and used in study of organisms. Likewise, unicellular, the multicellular organism shows varieties of cells as what we have previously seen. These are either sperm cells, red blood cells, white blood cells, muscle cells, nerve cells, or any other cell types which are connective tissue cells, or the subtypes cardiac muscle cells, or the gastric organ which is composed of some specialized striped muscle cells or smooth muscle cells. And these all the different types of cells have different functions as per the organ or the organism system. Each and every system of living being is composed of specialized cells, and every cell have their own different functions, and they work as per the organ or the tissues or the cells there. So basically we will see what is the general organization of prokaryotic cells. Morphologically point of view, if we study the prokaryotic cells, these are very primitive, or we can say having nuclear material as well, and therefore they are termed as prokaryon or prokaryotic. They occur in bacteria and blue-green algae or cyanobacteria. So these are the basics, and it consists of central nuclear components which are not bounded but scattered in the central area. Now works as a nucleus, generally called as DNA molecule, RNA molecule, and nucleoprotein material. These all the DNA, RNA and material, nucleomaterials or nucleoprotein materials are surrounded by a cytoplasmic ground substance, and in that ground substance this nucleomaterial is situated. Then this whole material along with DNA, RNA and ground material is surrounded by a thin membrane called as plasma membrane. This material bounds all the intracellular material and together called as prokaryotic cell. The cytoplasm of these prokaryotic cells lacks in well-defined cytoplasmic organelles such as endoplasmic reticulum, Golgi bodies, mitochondria, centrioles, lysosomes, etc. Now, what is the structure of this prokaryotic cell? We will study the example of bacteria, the famous Escherichia coli bacteria. This bacterial structure, if we will see, we can find out, we can understand easily what the structure of prokaryotic cell is. Now, Escherichia coli diagram is shown in the next slide, but prior to that we will see what is the bacteria. E. coli is a gram-negative bacillus, means gives negative staining to the cells, which is non-pathogenic, means not causing any harm to the human being or any other organism, and found in the mammalian digestive tract, means in our body these E. coli are naturally present and they perform the activities of metabolism and digestion. So it is measured about 2 micron long and 0.8 to 1 micron thick. It has outer cell wall of 100 degree angstrom thickness, and its cell wall and plasma membrane have typical structure of bacterial cells. The cytoplasm of this E. coli is colloidal in nature and containing roughly 5000 chemical compounds, means about 5000 chemical compounds ranging from water to DNA molecule. So it contains about 20,000 to 30,000 ribosomes, and these all are 70S types. Generally, ribosomes are of two types, that is 70S and 80S. But in prokaryotic cells, only these 70S type of ribosomes are there. And these are the main cellular organelles, because others are absent, we have seen in previous topic. So all genetical informations are transmitted or carried out by a single or double stranded circular chromosomes of DNA molecule. DNA of E. coli does not remain wrapped in histone proteins like the DNA of eukaryotic cells, but instead the nuclear region lacks any membrane in cytoplasm and therefore called as nucleoid, not nucleus. So therefore these are being known as nucleoid or nuclear material, not nucleus. They reproduce amitotically or asexually by, that is by binary fission. Simply it becomes two pieces of the cell and both pieces are working as a single cell and their broken boundary is regained or rejoined by its own cells by repair mechanism, and in this way the binary fission from one cell two cells are developed. So in this figure prokaryotic cell of E. coli, you can observe here that there is a outer cell wall. Then in outer cell wall, the respiratory chain enzymes are located in the inner wall. This inner wall is nothing but the plasma membrane. In between this cell wall and plasma membrane, these materials, respiratory chain enzymes are located. So in cytoplasm, or we can say matrix material inside the cell, you can see 70S ribosomes. 50S plus 30S both subunit, if you will join, the 70S ribosome is prepared. Then protein molecules are randomly scattered found in the cell material. Then DNA molecules are these are the coiled structures are the DNA molecules. They are randomly scattered. They are not aggregated in the center, not aggregated or bounded by the membrane. Therefore, we cannot call it as a nucleus. Instead, we know it as a, or we can call it as a nucleoid material. Then these are single stranded small pieces are nothing but the RNA, because DNA is double stranded and RNA is single stranded. This is very natural and basic. In spite of this all, the polysomes are there. These are the polysomes which are randomly moving here and attached at the both sides of the RNA material. This is the compound RNA material. Then this matrix in which all the materials are suspended are nothing but the cytoplasmic matrix, and in this way this prokaryotic cell is composed together as a E. coli bacteria, or we can say single unicellular cell. Now next to that we will see the detailed structure of the prokaryotic cell and its all components, its all working, its all functions, and then somehow how it works as a reproduction. So this topic will be further described in the next lecture.